Hello, can somebody confirm that you can hear me?
I can hear you.
Great,
We can hear you.
great. All right, let me state that I've got some great
Yes, can
done.
hear you.
Okay. Um, so let me start by saying I've got some grading done and then I'll share my screen. I've got some extra credit assignments done, like the extra credit picture I put in the grade, and I have not graded the extra credit study though. I have the extra credit study sheet graded. I think I graded that one earlier. I got worksheet lecture 6 graded. Started grading worksheet for lab 7. I'm only about halfway through that. I hopefully will finish that tonight. We'll see if I make it, but I think I'm halfway on grading that. I think that's it. Oh, I have quiz 1A graded, but that's also it. I do not have quiz 2A or quiz 3A graded. I need to go in and take a look at quiz 1B, quiz 2B, and see if I'm going to award any extra credit points. Let me see. All right. Any questions about the grading? Whoops, didn't mean to do that. Let's go back to that. I should have all of the unknown assignment out for last night, meaning if you requested a test and the incubation period was up last night, I sent out the results. I have not looked today, and I probably won't until fairly late tonight. If you're having questions about the unknown project, please ask. A number of you have not started the unknown project. You will need to get started on that pretty soon. If you wish to finish, this project does take some time. There are a few students, maybe one or two, that I think are finished, but they were very lucky and they only need to ask maybe like 2 tests to get their unknown identified. Most people will request, well, about 4 tests to identify their unknown. And some people, if you're very unlucky, you'll have to ask like 5 or 6 tests. All right, let's go ahead and share my screen now. Remember to take a look at your unknown project. Just scroll down to week 5 lab, and everything's in the unknown, about the unknown. I guess that should be unknown project, not unknown report, but you get the idea. And there's a sample journal for how your journal should more or less look. It is true that this was done in the lab, so there's some streaking out and stuff like that. You won't do that, but everything else after that should be similar. And then the unknown notes, where you're putting all this information for the unknown project. The table, the unknown list for the different species, and then the instructions. And of course, using a dichotomous plan. Remember, when you're working on the unknown project, you are supposed to be using a dichotomous game plan. And then there's a link for how the unknown report will be graded. All right. Let's go ahead and go to where we're supposed to be. So today is February 14th. And we're supposed to be starting chapter 8. I'm not sure we're there yet. I think we're supposed to be, I think we're on chapter 6. Let me check on that. Does anyone recall whether we finished chapter 6? I don't think we have. Well, further than slide 17. No, that's not where we are.
The last I have is a facultative anaerobe.
You what? The last thing that was
Facultative anaerobe.
facultative
That's the
anaerobe.
last note I took. Uh-huh.
Okay, yeah, I talked about oxygen. I remember that now. Come on, let's move. Yeah, we've got a lot of chapter 6 to go, so we'll be a little bit behind time. I thought I talked about media. Culture media is what we grow our bacteria in. Media is plural, medium is singular. We have a liquid media, which we call a broth, and you can add something to the liquid media to make it a solid, such as usually what we add is agar, but you could add other things such as starch. The media you would get if you actually went into the lab would be sterile. That means there's no living microbes in it. We call it an inoculum when we introduce microbes into the medium. And then when the microbes are growing in the culture medium, we call that a culture. We have a very wide variety of media that are available. If you were to come into the lab, I could show you a book talking about the different media we have available in microbiology, and it's thicker than this. It's about that thick. It's also bigger than that. And each page, there's one media. It just lists the ingredients in that medium. So there's a large number of culture media available to grow microbes. Media contain all the nutrients needed to grow microbes. There are different types of media. They all provide an energy source. Well, let me think about that. Unless it's something like a phototroph, in which case you don't need the energy source. Usually they have an energy source, a carbon source, sources of nitrogen, sulfur, phosphorus, sources of trace elements. And these are usually obtained just from either the water, or if you're grinding up part of a cow and putting it in the media, which would be called beef extract, that would have trace elements in it. And then if the organism needs it, we supply organic growth factors. If you might recall, we mentioned that one organic growth factor for humans would be vitamin C. So essentially, for humans, vitamins are all in an organic growth factor, but there are other organic growth factors. Like humans need the amino acid lysine. We cannot make it, in which case we call it an essential amino acid. It has to be supplied in the diet, but in generic terms, we'd call that an organic growth factor. And bacteria occasionally need organic growth factors like Streptococcus pyogenes, which causes strep throat, requires an organic growth factor, or maybe several. Agar is something we add as a solidifying agent for culture media. It's a complex polysaccharide derived from seaweed. It's usually found in a petri dish, but we can have it. Let me see if I can find this. We can't have it in a tube. There we go. In which case we have two types. Come on, mouse. sort of a tube. I'm not doing a real good job making a tube. If it's flat across, we call that a deep, D-E-E-P. And if it's not flat across, we call it a slant, like we could grow it, put the auger like that. And that's called the slant. Any questions about any of that? Generally, agar is not metabolized by microbes, and that's why it is frequently used to solidify a liquid media. There are different types of culture media. One type is a clinically defined media. Let me blow this up. Here are two clinically defined media where we know the exact composition of what is in the medium. Like this one, which is a glucose media, contains glucose, ammonium phosphate to supply nitrogen and phosphate, sodium chloride just to make the tonicity correct, magnesium sulfate, mainly to supply sulfate, but also magnesium, and potassium phosphate, mainly to supply phosphate, but also to supply potassium. And then you just add water, and that are all of the molecules that are in this medium. You can have more complex, like this has a whole bunch of different additives, and you would need to add more ingredients if you had a fastidious organism, something that required a lot of additives. Normally, we only use the chemically defined media for growing autotrophs because they are much easier to grow. They supply their own nutrients using just usually carbon dioxide and water. A fastidious organism is one that you have to add extra things to it, and we'll talk a little bit more about that. Most media we use in microbiology is a complex media, and it contains extracts and digests of yeast, meat, or plants. These items provide vitamins and organic growth factors, and the exact chemical composition is not known of these complex media. For example, a nutrient agar contains peptone, that's a partially digested protein. Beef extract, so you grind up the cow, sodium chloride to make the tonicity, and then agar just to make it solid. Oops, didn't mean to do that. Let's go back. The exact molecules in this media are not known because it'll depend on what molecules were in the cow. And that will depend on what the cow had been eating. So we don't know all of the molecules that are in this medium. Any questions about any of that? Peptone, a partial protein digest, is the primary energy source, but it also supplies carbon for carbon source, nitrogen and sulfur sources. And then beef extract does the same. It also provides energy, a carbon source, a nitrogen source, and a sulfur source. We use complex media to grow heterotrophs of bacteria and fungi that generally require an organic carbon source, which would be the beef and the peptone. And these guys are a little easier to grow because they can make a lot of their own molecules, but you do need to supply a carbon, a nitrogen, a sulfur source, and an energy source, which comes from the peptone and the beef extract. E. coli would be an example of a heterotroph. Let me go back just a moment. Something that is fastidious, you'd have to add more things than this to. A fastidious is an organism you have to add like organic growth factors to, and you might get a few organic growth factors from the beef extract, but you wouldn't get very much. And Streptococcus pyogenes, which causes strep throat, would not grow on nutrient agar. Okay, doesn't have the organic growth factors that it needs to grow. Then there's anaerobic media. This is a media for growing obligate anaerobes. They must avoid the exposure to oxygen. It has a reducing chemical in it, such as sodium thioglycolate, that combines with and removes oxygen. Therefore, there will be oxygen depletion, and we mentioned that in the thioglycolate tubes at the bottom of the tube, they have no oxygen, and then you can grow obligate anaerobes there. Any question about any of that? There are selective media. These media have chemicals in them that halt or inhibit the growth of a particular group of microorganisms while not affecting other groups of microorganisms. For example, we could have or add 7% sodium chloride to the media, and that actually we call it 7% sodium chloride media. This will only allow halophiles to grow, meaning salt lovers to grow. Most organisms would not grow in 7% sodium chloride, so they would be selected against. And this is a selective media. Any questions about that? We have other medias, a differential media, and this provides differences between two different types of organisms. The differences are not related to how well the organism grows on the medium. It may distinguish the colonies, they may look different or perform differently or just have a different color. An example of differential media is blood agar. It allows you to determine the hemolytic activity of the bacteria. So in this plate here, let me blow this up from that one, sorry. The bacteria is on the beta, the white beta, and then this clear region around it is where the blood cells have been lysed. So you can actually see through the media right there. That's actually clear, not white. Where the bacteria is growing, the bacteria are white. So that's where the streak the bacteria is. And then that's beta hemolysis or complete hemolysis. This is alpha hemolysis or partial hemolysis. Once again, the off-white color is where the bacteria is streaked in an alpha. And you see this dark ring around the alpha as well as inside the alpha. That is from the partial lysis of the blood, making the bacteria look either dark in this case, or sometimes it looks greenish. And that's from alpha hemolysis or partial hemolysis. And then here we have gamma hemolysis. Once again, the bacteria is streaked white, and we see the red blood cells come all the way up to where the bacteria are. That is gamma hemolysis or no hemolysis. Any questions about any of that? Sometimes you can have the media types together, like selective and differential media. It is both selective and differential. Eosine methylene blue or EMB plates are an example of a selective and differential media. It's selective in that it selects against gram-positive bacteria from growing on the plate. So if we have growth, then it's most likely gram-negative. And then it's differential on the basis of lactose fermentation. If it ferments lactose, it'll either be dark, like this, or look metallic greenish, kind of like that. This right here ferments the sugar lactose. And like I said, it's either dark or greenish looking, metallic greenish looking. And then over here, you can see the color best. In some of these, you can see it. This does not ferment the sugar lactose. It is a gram negative, so it can grow on EMB. But because it doesn't ferment the sugar lactose, this is actually white, which is visible there. These blackish, and that's not totally blackish. It's just the way that the photograph angle is appearing. It's not black. We have another type of media, which is called an enrichment media. It encouraged the growth of certain organisms to grow. So therefore, it is selective because it selects against other organisms from growing. But it is not used like a selective media. We grow it to enrich for bacteria that we want to see. And we use it to increase the numbers of the desired microorganisms to particular detectable levels. For example, if we were going to streak a patient's fecal sample or maybe bacteria from the soil, there's going to be millions of other bacteria in the streak. And if the bacteria we were trying to find is rare, then you would be unlikely to find it from a culture streak. So it's particularly useful to grow that sample in an enrichment media and then enrich for the sample that you're looking for. And let me see if I can talk about that a little bit better. Suppose we're looking for a phenol degrading bacteria, but yet there's thousands of other bacteria in the soil. If you were to streak it out and you had 10 isolated colonies, the chance of getting a phenol degrading bacteria would be really rare, meaning unlikely to get it. If you were to grow it in an enrichment media, you could select for phenol degrading bacteria. One way to do that would be make phenol the only carbon and energy source. That would select for phenol degrading bacteria. And that would increase the phenol degrading bacteria's number in the culture. Even if it only increases it 10 times, you could take 1 mil out of that sample from the enrichment media and put it in a new enrichment media. Now we've enriched it 100 times. And then to enrich it 1,000 times, you can transfer once again. And I'm just assuming that you will enrich it 10 times each time. If it was like 1 in a million and you enriched it 1,000 times, you've made it so that you will get 1 in 100, 1 in 1,000 colonies that you sample would be a phenol degrading bacteria. You'd probably want to enrich it further because the minimum you need to get it would be 1 in 100. And to tell you the truth, what we would like to have is to enrich it up to 1 in 10. Then you only need to streak out 1 plate. If you had 10 isolated colonies, 1 of them would be the sample you're looking for. Any questions about enrichment and how we go about doing it? All right, so this slide is showing you a summary of the different media I discussed. I'm going to skip it because we're a little behind schedule. A pure culture is a culture that contains only one species. How we get that is to streak out on a plate and get an isolated colony, take cells from that plate and streak it out on another plate to get isolated colonies. So we pass the cells through 2 colony formation events. That makes the chance of that being a pure culture greater than 95%. If you were to only streak it out 1 time, the chance of getting a pure culture is only about 70%. And that's just not good enough when you're working in microbiology. Okay, so you do want to streak it out 2 times and pass the cells through 2 isolated colonies. Because when you streak out on a plate and you get an isolated colony, you don't know if that colony was formed from 1 cell or 2 cells or a bunch of cells clumped together. So you do need to streak it out 2 times to ensure that you will have, well, it doesn't even ensure that you'll have a pure culture, but it will increase the chance that you have a pure culture to greater than 95%. How you streak out a plate is shown in this slide. What you do, can you see if I streak? Oh, you probably can't see there. Let me hold it back here. What you do is you streak out on a plate. And this isn't the best streaking, but it's good enough. What you do is you streak out, come on, mouse, in sector 1, 20 to 30 times in sector 1. This is a little short, but it's trying to show you what the streaking looks like. And then you sterilize the loop. So I'll put that in the Bacterial incinerator. And then you streak out sector 2. Now in sector 2, you streak out in sector 2, and then you go into sector 1 to pick up cells on your loop. Come on, mouse. So here we're picking up cells from sector 1 and then carrying them back into sector 2. Ideally, you'd only want to go into sector 1 about 3 times, maybe 4 times. Sorry, my mouse keeps dying. 1, 2, 3. So you'd want to stop right there. And actually, if they would stop there, that would be better. You want to do it when in sector 1, the cells are somewhat diluted. Here, they're very concentrated, the cells. And then down in this region, they're more diluted. You want to grab from sector 1 where they're more diluted. You'd never want to grab in this region where they're highly concentrated, because you want sector 2 to be more diluted than sector 1. And here in sector 3, it's showing it a little better. You streak out sector 2 then. And then you streak out sector 3. Once again, you take your loop, pass it in the Bacterial incinerator, sterilizing what's on the loop, and then come back to sector 3, which should be over here, and then go into sector 3 where the cells are somewhat diluted. And this is going a little too far. They should have stopped about there. And then back into sector 3. And then you should come back to sector 2 about 3 times. Okay, to make sure you pick up cells on the loop. For some reason in sector 3, this is only showing you 1 time. And that's just the picture we have, so I use it. Any question about any of that? Now, this streaking for colony isolation, go on. Go on. All right. If you have a question, go ahead and ask. This procedure only works well when the culture that you're looking for happens to be present in your starting sample at a fairly high percentage of the total microbial population. So if it's less than 1 in 10 of the cells, trying to obtain what you're looking for would just be too difficult. Okay, even 1 in 10, we have a little more than 10 isolated colonies here, but you want it at least to be 1 in 10. And that way you're only looking at 10 colonies to try and get the culture you're interested in. If it's lower than that, then you need to enrich for it. Okay, does anyone have a question before I move on from this slide? All right. Remember that bacteria do grow, and by that we mean they divide, most bacteria grow by binary division, where the cell literally splits in two. But there are a few bacteria that grow by budding, and that is where the parent cell, I'll make it this way, puts out a little bud, and then that bud grows with time, and it eventually will separate. It usually separates when the bud approaches the size of the parent cell. And there are some bacteria that also reproduce by forming reproductive spores. They usually form a reproductive spore at the tip of a filament. Let me find paint again, and I can illustrate that. There it is. Come on. And I'm just going to draw it with one cell, but this could be like a bunch of cells growing like this. I'm just going to make this the parent cell. And then it makes the reproductive spores. The reproductive spores are little cells on this cell. And it's reproduction because the parent cell is making more than one reproductive spore. It's usually about 4 to 6, something like that. And they're usually at the tip, and that's so that with the wind, these reproductive spores can go elsewhere, or maybe an animal comes by and picks up the spore and carries it elsewhere. And then that spore can grow into essentially another parent cell. Okay. This is reproduction because, like I said, that one cell makes more than one cell, and usually that parent cell doesn't die in a reproductive spore. That's different from an endospore, which one parent cell makes one endospore, and then the parent cell dies. So that makes one, making one. That's not reproduction, an endospore. But reproductive spores are reproduction. Only a few cells form reproductive spores, and only a few bud. Here you're seeing binary fission. What happens is the DNA of the molecule divides first. The DNA then moves towards opposite ends of the cell. And then the cell literally grows a cell wall and a cell membrane in between the DNA, copies of the DNA. Okay. And then the cell splits into two. And then we have two daughter cells from one parent cell by binary fission. And let me see if I can blow that up. This is actually an electron microscopic image where this cell is getting ready to divide. And there you can see it's starting to form the cell wall and the cell membrane in between the DNA here and the DNA there. Most bacteria do grow by binary fission. A few grow by budding. Let me see if I can get this to work. This is showing you budding in yeast, but it doesn't matter because, anyone see a start button? Oh, there it is. So this cell is starting to form a bud there. And there's the bud now. If I can blow that up, you can sort of see the DNA moving into, into the cell. Now let me clear that and do that over again because I had a message on there. It's saying my internet's connection is unstable. Let me make sure there's nothing between me and the computer. There the DNA moved in. And there the parent cell is going to make another bud here. And this cell is going to make start putting out a bud there. Any question about budding? Let me go a little further here. About right there is where the bud separated from its parent, meaning it formed a cell wall in between the two cells. When we're talking about bacteria and growth, it's good to use generation time. The generation time is the time required for a cell to divide, and it is the same time for the population of these cells to double. So the doubling time is the same as the generation time. Now, the generation time, we can be in bacteria as fast as 20 minutes, like E. coli, may, I guess would be a better way to say, may reproduce every 20 minutes. Most bacteria take a little longer than that. Most bacteria grow or divide in 1 to 3 hours, meaning it takes 1 to 3 hours for one cell to become 2 cells, or the population size to double. Now, when we're talking about the generation time, we might say 20 minutes is the generation time, but it's understood that it's 20 minutes per generation. That per generation is understood, okay? So the generation time is the time required per generation. And don't get that mixed up, because we're always used to talking about like something like miles per hour. The generation time is not, I don't know, the generations per hour. It is the time per generation. Now, there are some bacteria that are extremely slow, like Mycobacterium leprae takes 10 days to double. Most of the mycobacterium take longer than most other cells to grow because they put mycolic acid in their cell wall. And it takes the cell a long time to make mycolic acid. It's a big, complex molecule. And Mycobacterium leprae, it used to be the slowest growing bacteria we know. I don't know if it still is or is not. Like I said, in the old days, I used to know, but I've been teaching microbiology now for, gosh, probably about 20 years. So I'm not sure if Mycobacterium leprae is still the slowest bacteria we know of, but its doubling time or generation time is about 10 days. When we're talking about the generation time, microbiologists like to use semi-log plots, and that means it's a log on the number of cells, but it's actually arithmetic on the number of generations, meaning the x-axis is a regular arithmetic scale, but the y-axis is a log scale. If we were to use an arithmetic scale on the y-axis, then the graph of the growth of the bacteria would look something like this. There would be a whole bunch of plots, points down here, like one cell would divide into 2, 2 cells into 4, 4 into 8, 8 into 16, 16 into 32. So there's a whole bunch of points here. And then this point is at the end of the graph. The next point would be on the ceiling. You can't get the next point anywhere on the graph, on the arithmetic scale. And then the function here is a rather complex function. It would be something like y equals, I'm going to say n x squared plus mx plus b, meaning it's a complicated formula for the growth. On the other hand, if we're using a semi-log plot, we scatter out the points here so that we can actually put them in the graph. And then the next point right here is actually going to be something like right here. It won't be on the ceiling. It might even be on the piece of paper, okay? So we can put more points on the graph, and then it scatters out the points down in this region, and then we can get one more point on the graph fairly easily. And then the equation is very simple. This equation for the growth is y equals mx plus b. And mathematicians and the biologists always like using a simpler equation to explain the growth. So that's why we use a semi-log plot when we're talking about the growth of bacteria. When we're talking about the phases of growth, it doesn't really matter what you're talking about. We're going to talk about it with microbes, but this actually has been seen in animals, where somebody introduced a bunch of deer in the island and the growth curve looked like this. Okay. Initially, when you inoculate bacteria from one environment into another, like from a solid media to a liquid broth, there will be a lag phase. What's happening in the lag phase is the bacteria are not growing because they're making the enzymes they need to grow in the new environment. And once they have those enzymes made, they can then start growing. Once the bacteria start growing, they enter the log or exponential growth phase because they're growing exponentially. That growth will slow because initially in the exponential phase, there's few bacteria, lots of resources, and lots of space. But up here, there's fewer resources, fewer space, and there's more waste products being built. And of course, initially there are no waste products. So there's low waste products down here. And then up here, there's high waste products. So the bacteria begin to slow their growth. Eventually, they'll enter the stationary phase where the bacteria population number stays about the same, what's called the stationary phase. The lucky bacteria cells have some nutrients around them, some space, and not too much waste products around them, so they will be growing. The unlucky bacteria cells will have very little space around them, very few nutrients around them, and lots of waste products around them. So they will not be growing and may even be dying. In the stationary phase, you can say that growth equals death. And that's why the population number stays the same. Growth equals death. But eventually, more and more of the media will be used up. The waste products will increase in number. The cells, well, the cell space will maybe increase in number, depending how much that happens and how quickly the cells die, I guess. If the cells exactly equal, the death equals the growth, actually the space would be the same. Anyways, the resources are being used up and the wastes are accumulated. Now the cells dying outnumber the cells growing. And then later in the death or decline phase, where the population number starts declining, we will only have cells dying. There will be no cells growing. And that's because the nutrients they're used up. The waste products will be very high too. How quickly the population goes from, I don't know, their highest number to zero will depend on the bacteria. If the bacteria form endospores, this can last for centuries, the decline phase. if the bacteria is something that dies fairly easily, like E. coli, we're talking about it goes from here to there in about a week or two. If you don't hear me, just ask me to repeat something because I continually am getting messages that the internet connection is unstable. So the 4 growth phases are the lag phase, the log phase, the stationary phase, and then the death or decline phase. All populations go through this, and that's because when you introduce a population into a region, it starts growing, and then that starts using up the resources. And then the population enters the decline phase, assuming that the resources are limited. Will the human population ever approach this? We will see. Hopefully, if it does, the death or decline phase will reduce the population numbers to a point where then we can stabilize the population. I mean, hopefully the human population wouldn't starve to death, but we'll see what happens. Like I said, they've seen this growth happen when they introduced deer to an island. The deer numbers just increased, and then there are too many deer on the island for the, I don't know, trees or life on the island to support the deer population, and the deer population declined. There are a number of different ways to measure the growth of microbial populations. There's direct methods where you directly count the bacteria cells, like the plate count, the filtration method, the most probable number method, and the direct microscopic count. There are also indirect methods such as turbidity, metabolic activity, and then the dry weight. I've used all of these methods for getting an estimate of the number of bacteria, except for the most probable number. I've never used that one. In the plate count, it is the most common method used in microbiology for obtaining the measure of bacteria in a solution. It is also called the viable plate count because you only count viable cells by this method. And this method is mainly the method used to determine how many viable cells are in a solution. The results are often reported as colony forming units because we count the number of colonies that form, and we do not know if a colony was formed by 1 cell, 2 cells, or by a clump of cells. So we call it the number of colony forming units per mil rather than the number of cells per mil. Any question about that? To obtain a plate count, you first make a serial dilution of your original sample, and then you inoculate each of these dilutions onto a plate. And then you count the colonies on the plates where there's a total between 25 and 250 colonies on the plate. I'll talk about why we do that in a minute. And then you base the count on the dilution to get an estimate of the number of bacteria in the original sample. And we'll skip that. So you have your original sample, which isn't shown, and then you make a dilution. You might take 1 ml and dilute it into 9 mL, so that's 1 to 10. And then you mix that up, take 1 ml here and put it into 9 mL of water or broth. And now you have another 1 to 10 dilution. 1 to 10 and 1 to 10 is 1 to 100. You mix that up, take 1 ml, and then you get your 1 to 1,000 dilution. Mix that up, take 1 ml, then you get your 1 to 10,000, do the same, and then you get your 1 to 100,000. You then plate each of these onto a plate. There's two types to do that. You could do the pour plate method, or you could do the spread plate method. Either way, you put a sample on the plate, and then you spread the sample out, maybe with a spreader, or with, if it's a large enough volume, you just tilt the plate and that sample itself will spread on the plate. Then you incubate these plates, usually for about a day, and then you count the colonies. What you'll get is something like this. Most of the dilutions will have too many cells to count, like the 1 in 10 dilution, the 1 in 100 dilution, the 1 in 1,000 dilution, in which case you go something like 2n Tn Tc, meaning too numerous to count for these plates. This one we can count, and this one we can count. We don't use this one. We use when the colony numbers are between 25 and 250, because the mean here, let's say this is 25. No, let's say it's 40 because that's 4. The mean here would be something like 40 plus or minus, let's say 40 plus or minus 1, which is plus or minus less than 3%. Here, our mean is 4, and that's plus or minus 1. And that would be 4 plus or minus 1, or plus or minus 25%. And that's just not a very accurate mean if we base our colony count, come on mouse, on this plate. So we base the colony count on this one. Any question about that? All right, let me go back a minute. Oh, come on. The point is here, we're only getting colonies from viable cells here. So we're only counting viable cells in the original solution. And let's say it's 40. I'm not going to do a real count. I'm just saying it's about 40 here. And the dilution is 1 in 10,000. So that would be 40 times 10,000. What would that be? 400,000 cells per mil are in our original inoculum. And if I did that wrong, you understand about what I'm trying to do. You multiply the count here times the dilution to get the number of cells per mil in your original sample. Another way to do it is the filtration method. We use the filtration method when the bacteria are rare in number and you could not get accurate counts or good reliable counts by doing the viable plate count method. So like when we're sampling drinking water for bacteria, they'll put it through a filter and then put the filter on a plate of agar, and then you just allow the colonies to grow on the filter on the agar. And then you just count the number of colonies here. So that's the filtration method. It's only used usually when you're counting cells when they're very low in number, and then you pass it through a filter. Like for drinking water, you might pass a whole gallon through the filter before you then get it to grow, because you don't expect very many cells in even a gallon of drinking water. Now, the bad thing about the filter, you have to filter either a liquid or air through the filter. So the bacteria have to either be in the air or in a liquid. A direct microscopic count is where you put your cells on a slide, which has an exact measured volume under the slide. Cells are then counted microscopically in the microscope. This is not good for motile bacteria because they move around. And it's difficult if the bacteria have other issues like a capsule around them. Not impossible, but it's difficult. Like I said, you use a special gridded slide that has a fixed volume underneath the slide. And usually the volume will be however many cells you have in this one square here. You do your count here, and that would be about 20, maybe 15. And then you multiply by 1,000,000, meaning that's the volume there, 1 microliter under this. And then that would be, let's say it's 15 times 10 to the 6, because we're multiplying by 1,000,000. So 15 times 10 to the 6 cells per mil in your original sample. Did I say that right? 15 times 10 to the 6 cells per mil in the original sample. The hard part here is somebody actually has to go on the microscope and do the count, but it's fairly quick. It would be impossible to do this count, though, if the cells are motile, and that's because this cell could move there, and these two cells could move there. And one disadvantage of all of the methods, except for the viable plate count and the filtration, is you're going to be counting dead cells, because it's impossible to tell, is that a viable cell or a dead cell? And we just can't tell by looking. And that's true of all the methods except for the viable plate count and the filtration method. We can also determine the microbial numbers by indirect methods, such as turbidity, metabolic activity, and dry weight. In turbidity, what you do is you shine light through the media, and then you notice how much light is absorbed or transmitted through the media. And then you shine light through the media containing cells, and less light will get through because the cells will block the light coming through. And then you measure the amount of light coming, and then you compare that to a chart. So somebody has to make the chart for how many bacteria make this concentration of light. And then on the chart, you'd have how many bacteria per mil to make a less light get through, and then how many bacteria in the solution to make more light get through. And then at the very start, you'd have just the media itself, how much light gets through the media. So you have to look on the chart, but once you do that, once the chart is developed, you can keep it forever, certainly for the lifetime of the lab. And as long as you're using that same media and the same turbidity meter, you can use that chart forever. And what you do is you then just take your sample and see how much light has come through, and then look up on the chart how many cells per mil this is. And this is a very quick way to get the estimate of the number of cells. Like I said, one disadvantage of this is we can't tell if these cells are alive or dead. And it does count both living and dead cells. All right, the last two ways we need to talk about are you can determine the number of bacteria from their metabolic activity, like you could measure the amount of CO2 that accumulates. And generally, the more bacteria are present, the more CO2 will accumulate. And then you make a chart of that and determine how many cells are there, depending on the metabolic activity, like the amount of CO2 produced. You can also do something similar, and that is take out the cells, meaning centrifuge them from their solution, let them dry, air dry them, and then weigh them. One advantage of this is you can weigh them at your leisure. It doesn't have to be that day. And then the more bacteria there are, the more the weight will be. And then you just look on the chart saying that, I don't know, 1 gram equals so many bacteria. Any question about the methods we use for estimating bacterial numbers? All right, if not, let's begin chapter 8. As is always, I start the lesson with a lecture showing you the major goals and a rough lesson outline. So know the terms of this chapter. We'll talk about these terms and then be able to describe DNA replication, RNA transcription, and protein translation. And then lastly, understand the different types of mutation. We're going to talk about the structure and function of the genetic material. We'll talk about the flow of genetic information. We'll go into DNA replication, talk about RNA transcription and protein translation. Then we'll move to the regulation of bacterial gene expression, talking about the operons. And then we'll talk about mutation, the genetic transfer and recombination. And then lastly, we'll talk about genes and evolution. Any question about what we're doing? I think I have 20 minutes left. Let me make sure that's the case. Yeah. So genetics is the study of what genes are, how they carry information, how that information is expressed, and how genes are replicated. A gene is a segment of DNA that encodes a functional product. Usually, a protein is encoded by a gene, but there are a few genes that do not encode protein. These genes encode RNA, and that would be the mostly the rRNA genes and the tRNA genes. Any question about that? So most genes do code for a protein, but not all genes do. The genome is all of the genetic material in a cell. And because we're talking about mostly bacteria, we're talking about all of the genes in the organism. The study of genomes is called genomics. A genotype is the genes of an organism. For example, if you have blue eyes, your genotype is lowercase b, meaning we have, because got blue eyes, we have 2 lowercase b genes, and I could say alleles. That differs in the case of a brown-eyed individual. The brown-eyed genotype is either capital B, lowercase b, or capital B, capital B. The point is that's the genotype. The phenotype is the expression of the genes. So in people with blue eyes, you have 2 lowercase b, lowercase b's, and then the eye color will be blue, the expression of the genes. In the case of those individuals with genotype capital B, lowercase b, or those individuals with capital B genotypes, they will have a phenotype of brown eyes. Any question about any of that? All right, no questions. Let's move on. Here we're looking at a chromosome to E. coli. E. coli chromosomes, all it is the DNA molecule with proteins in the DNA. and you can't look here and distinguish is this DNA or protein, but it's a very long string, and there it is. A chromosome contains all of the genes, and they're just a certain region of the chromosome, like right there, there's probably one gene, and then right there might be another gene. We'll use the right-hand one. Remember, a chromosome is both DNA and protein, even in prokaryotes. In eukaryotes, it is protein and DNA, but the number one protein is the histone protein. And you don't have histones in prokaryotes. Excuse me, you do not have histones in bacteria. I think archaea do have histone proteins. DNA is a polymer of 4 nucleotides. The nucleotides adenine, thymine, cytosine, and guanine. And you may learn them by these names, or you can call them A, T, C, and G. I don't care which way you know, just know one or the other. DNA is a double helix with proteins. And here we're looking at the double helix, meaning it has 2 strands of DNA. The strand of DNA, meaning one strand of DNA, is held together by the sugar phosphate bonds. So we have sugar here, there's the phosphate, there's the sugar. So the sugar phosphate bonds hold one strand of DNA together. And this is a very strong bond, that's a covalent bond. The backbone is called the sugar phosphate backbone. The 2 strands are held together by the bases only by hydrogen bonds. And the hydrogen bonds are very weak, like this bond breaks and reforms all the time. And the reason why DNA is held together in our cells is because when this bond breaks, all the other, well, not all the other, but most of the other hydrogen bonds are formed and they hold the DNA together. And then this will reform and maybe that bond will break. The 2 strands are held together by hydrogen bonds between A and T and C and G. A and T are complementary, so they'll pair. And then C and G are complementary. Do note that A and T have 2 hydrogen bonds, shown here and here, and C and G have 3 hydrogen bonds. And then lastly, let me state that the 2 strands of DNA are anti-parallel. That means one strand runs this way, the other strand runs that way. Okay, and I would kind of like to do it that way, but I can put my hands better that way. Any question about that? Not sure why that popped up. Okay, within a cell, the DNA flows in what we call the vertical flow of DNA within the cell, from DNA to RNA to protein. So the DNA, the information in the DNA is encoded by DNA, and that information is transcribed into RNA, and that information in RNA is translated into the protein. And this is known as the central dogma in biology. This is what we used to call a law of biology, meaning that DNA flows from, the information in DNA flows from DNA to RNA to protein. And by law, it means that the DNA information doesn't flow any other way, which in our cells that are not infected with a virus, this law is true. The information flows from DNA to RNA to protein. Now, this law is not absolute in the sense that with some viruses, information can flow from RNA to DNA, and we'll talk about that later in the term. So you don't need to know about that now, but we will talk about that. The flow of genetic information within the cell happens that way, but we can have the flow of genetic information flow between generations of cells, where we have a parent cell becoming two daughter cells. The parent cell will replicate its chromosome. Each chromosome will move towards opposite sides of the cell, and then the cell will divide down the middle by binary fission. And this is called the flow of information between generations of cell, meaning the information flows from DNA in the parent cell to the DNA in both of the daughter cells. And that we call the vertical transmission. The DNA also flows between cells of the same generation. Here, the DNA usually comes out of the parent cell, usually when the parent cell dies, but we're not going to talk about that. And then the DNA is outside the cell, and then it comes to another cell called the recombinant cell, which takes up this DNA that came out of that cell. And then this cell gains DNA from another cell. I've altered the figure in the book to make this a little more clear what's going on with the flow of DNA between cells of the same generation. Any question about any of that? So the flow of generation, the flow of genetic information can happen three ways: within a cell, between generations of cells, and then between cells of the same generation. Now, in humans, we have that information happening. We also have this one happening, but in which case DNA flows from two parents, generally to one offspring. I suppose it's theoretically possible we could have this happening in human cells, but this essentially never happens in human cells. I should say naturally. I suppose in the lab you could make it happen. All right, when we're talking about the vertical flow of information within a cell, the information flows from DNA to RNA to protein. The language of the DNA is nucleotides, and there's 4 letters in the alphabet, meaning there's 4 different nucleotides. The language of RNA is also nucleotides, and there are also 4 letters in the alphabet of the RNA nucleotides. And then the language of the protein are amino acids, and there are 20 different amino acids, at least in most living organisms like humans. We have 20 different amino acids. That differs from English, which has 26 letters in the alphabet. So you could say that the information in DNA is, that is a dog. The information in RNA is the same, but it's a different dialect. And you'd say something like, that is a dog. And then the information in the protein would be something like, quello è un cane, meaning the information would be in a totally different language. Any question about the vertical flow of information within a cell? All right, let's talk about the flow of genetic information between cells. This is known as the vertical flow of information. This happens when DNA is replicated. Let me blow this up. DNA replication is semi-conservative, meaning the 2 strands separate. We'll talk about that. DNA helicase does that, separating the two strands of DNA. And then one strand is used as a template to make a new strand. And that's true of this one too. The new strand is using this as a template to make the new strand. How that happens is the DNA polymerase will bind to this strand. Come on, mouse. See that there's a T here, so the DNA polymerase will add an A here. And then the DNA polymerase will move down. See, there's another T here, and it'll add an A. And it'll move down and see a G here, it'll add a C, etc. By DNA being semi-conservative, we mean if the original DNA is 2 dark strands, then each of the offspring will get 1 dark strand, the original DNA, and then 1 new strand, the white strand of DNA. Semi-conservative. How this happens is the DNA polymerase will add a nucleotide, and it adds a nucleotide triphosphate. When 2 of the phosphates come off, that releases the energy needed to bind this nucleotide onto the growing strand of DNA and then form the covalent bond between this nucleotide and that strand of DNA, or the growing strand of DNA. Any question about any of that? All right, so let's watch a little video on the replication of DNA.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases 1 by 1 in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about that? If not, I think I'll stop. Oops.
DNA sequencing is the process of working out the order of the building blocks, or bases, in a strand of DNA. Before we can sequence the DNA,
Any questions about any of that? If not, I'm going to log off here and we'll continue with this next time. All right. Goodbye.